Okay, I think I can quickly present my screen and just give a quick introduction.
Please.
Yes. Okay, can you see my screen now?
Yes.
Yes. So hello everyone. Welcome to the first session of Horizon. We are Jarurat Care Foundation. We are a patient advocacy group dedicated towards biliary tract cancer in India. We laid our foundation last year in March 2024, and ever since then we have made striking strides in the field of treatment navigation, preventative awareness, and community building, and also in field, research. We're working with different doctors. So we started our journey last year in March, stemming from my own personal cause where I lost my mother to biliary tract cancer. And that's when we decided on a mission to embark and set Zarurat Care Foundation as a one-stop shop for biliary tract cancer in India. And ever since then, we've been very fortunate to have support of multiple people. Like Dr. Javle and Dr. Sevanti in this journey, where we have been working closely with a group of oncologists to work on different research priorities. We work on treatment navigation. That is our passion project, which we started off with. We help the families who are battling with biliary tract cancer, cholangiocarcinoma, and gallbladder cancer with all the kinds of treatment navigation, anything beyond the hospital. We have so far managed to help around 250 plus families who've been actively battling this disease in India. And we try to help the caregivers as the point of contact, and also the families in every way possible with every kind of resources. We are currently also in the process of building our own AI chatbot, which can self-serve them, the audiences in India. We have also been working on the preventative awareness and awareness aspect, and for which we have worked on multiple webinars, which you can find on our social media handles, where we have often spoken about different kinds of, the reasons why biliary tract cancer is prevalent in India, what can be done, any kind of early detection methodology that can be used. Our we also, I'll try to make it quick. The current project line which we have been working on is, we are currently working on setting up a project which can help in the early detection screening of gallbladder cancer in India. So this is something which we are trying to work on, and it's still a work in progress with the AI screening work. The second thing we are actively looking forward to work on is understanding the genomics of biliary tract cancer in India, how it is different from the Western countries, and how we can further improve the prognosis of patients in India. So this is where we are. We are collectively working with patients as well as oncologists and researchers across the world. We are very grateful for everyone's support, and we would be delighted to have further support. If in case you would want to be connected with us, I'll drop in my email ID on the chat box and you can reach out to me. With this, I think I'll give it off to Dr. Javle and Dr. Sevanti to give opening remarks for Horizon. Thank you.
Thank you, Priyanka, for doing what you do. Congratulations on really organizing and doing a stellar job at getting all of us together. You are young, vibrant, and an inspiration, truly. You worked so hard to make this evening possible. And we are full of gratitude for you to really champion this cause despite your personal loss. To turn one's pain and agony into a force is what we are all about. I think a lot of us have gotten together here to project that cause. We are together here in propelling the cause. We are also together here in raising awareness. We are together here in finding a solution. I am full of gratitude to Dr. Milind Javle for helping us and for leading from the front, leading with an example, making time every time we have reached out. Priyanka has reached out, I have reached out, and also coming forward with guidance and strategic hand-holding to make this evening a possibility. Horizon this evening represents cholangiocarcinoma, gallbladder cancer. Today's discussion has a multidisciplinary projection of discussion of the subject, and also therapeutic strategy finding in a resource limited nation. But Horizon is not just about this. Horizon is envisioned as a global effort in bringing expertise, not just from MD Anderson, not just from global experts. We have Dr. Shroff today with us, not just from all across the other countries, but from tier 2, tier 3 cities and from urban and rural India. The goal is to bring together different parts of the world to champion the cause. And the cause is very much cholangiocarcinoma and gallbladder cancer, but also way beyond that to represent all malignancies, to represent multidisciplinary cancer care, to represent the cause of bringing clinical trials to India, and to represent making novel drug development a reality in the Indian society. With this,
I'm gonna deny
I
it.
and leave the stage to Dr. Milind Javle, Professor Milind Javle here, who has truly been pivotal in bringing this together. And without his leadership and support, this evening and the cause would not have been possible. Over to you, Priyanka, to introduce Dr. Javle.
Yeah, I would like to. I mean, more than grateful for Dr. Javle because he has been there since the beginning of our journey. I reached out to him just to ask for my mother's treatment, reviewing her reports back in 2023, and I didn't know that we would go so long in our journey. And I'm very grateful for his expertise for a person like him who's doing such a great work in the field of biliary tract cancer. So I'll give it off to Dr. Javle to speak about the horizon now.
Thank you very much, Priyanka. I would also say a quick word about both Priyanka and Semanti. Priyanka said she wants to start a foundation, and I had no idea she would carry it forward so effectively. And these are young people, IIT graduates, and, all kinds of fancy positions that they hold. And I asked her, would you consider going to Harvard for MBA or Stanford? She says it's a waste of time, really. So they come from this background where the sky is the limit. And then we have Semanti, the great star, who really opened up all the doors for us. But this evening in India, really belongs to Dr. Shroff. It is my distinct privilege to introduce Dr. Shroff, who will be our first keynote speaker for the monthly seminar series. And I was reading her short bio, which her assistant sent me, and it was quite remarkable what she has achieved in just 7 years since her fellowship. So Rachna is currently the Chief of Hematology Oncology and the leader of the University of Arizona Cancer Center. She's Associate Director of the Clinical Investigations Group at Arizona Cancer Center and a professor in cancer biology and a medical director of the Arizona Clinical Trials Network. She graduated from Brown University in Providence, then Jefferson Medical College, Barnes Jewish, where she did her residency. And of course, the peak to her career was University of Texas MD Anderson, right, where she did her fellowship. And I had the distinct privilege of being one of her professors when she was there. So Rachna has has achieved a lot in the short time. But relevant to this group, I want to say that she spearheaded the SWOG 1815 phase 3 clinical trial, which was the first phase 3 clinical trial in biliary tract cancer in the United States, enrolling about 450 patients to Gemcis versus Gemcis and Abraxane. And this study, you will hear the results from her. You always learn from studies, whether they're positive or negative. And I can tell you this combination is still being utilized, particularly in India. So Rachna, again, I don't want to spend any more time. The floor is yours, and I look forward to learning from your experience in biliary tract cancer.
Thank you so much, Dr. Javle, and thank you to Priyanka and to Sevanti and to the entire foundation for the invitation. It's such an honor and honestly quite humbling to be speaking. Number one, of course, with Dr. Javle, who has been one of my mentors and guides through the journey of biliary tract cancers, but also to this audience who I know, given the incidence of biliary tract cancers and just the incredible work that's happening in India, I think it's just so wonderful for you all to bring us together, and it is an honor to speak to you today. So I was asked to give a little bit of an update on what's happening in this space. And, I think I don't need to tell this audience that there is much happening. And what is really remarkable, is the honest truth is in the 15 years since I finished my fellowship at MD Anderson to now, Dr. Javle and so many others on this call and that I have the privilege of working with have absolutely changed the face of this disease, which I think is really remarkable because the other disease I treat is pancreas cancer. And yes, while we've made some progress, the absolute change in landscape for biliary tract cancers in the short time that I've been doing this has been so much fun to just be a very small part of. So these are my disclosures. I'm going to start with just, I think a slide that beautifully summarizes this. And this is just, from a US FDA approval perspective, which of course I think further doesn't really kind of understate exactly the progress that's been made. But, we've all known since the day I started treating this that gemcitabine and cisplatin has been really the backbone of our treatment. And that became standard of care back around 2009, 2010 when the New England Journal article came out. And then really since then, there was a slight gap. But then as you can see, the momentum and just the colossal number of drugs that became available to us in the last literally 5 to 10 years has been incredible. And some of them have been tumor agnostic approvals. Some of them have been very specific to cholangiocarcinoma, for instance. And some of them have in fact changed how we start with treatment. And so I'll start there, obviously, and just review some of the basic data. And please just remember that this is supposed to be interactive, and feel free to ask me questions. I know I don't need to belabor the TOPAZ study, but I really think of TOPAZ as a big turning point in the treatment approach to biliary tract cancers, and I'm sure you all would agree. This was the large global study, 685 patients that involved newly diagnosed metastatic or locally advanced biliary tract cancers, and it looked at Gemcis plus durvalumab, so chemotherapy with immunotherapy versus Gemcis and placebo. And this study, as a reminder, after 6 months of Gemcis with durvalumab, there was an option for, if you were on study still, to move to maintenance durvalumab versus maintenance placebo, with a primary endpoint of overall survival. So what I think is really just remarkable is that this was there on the left is kind of what we saw initially, and then we've seen the follow-up since. And so I think what's important to underscore here is that gemcitabine plus cisplatin plus durvalumab did improve median overall survival. But the separation of the curve and what we look at as the 3-year landmark overall at the 3-year OS analysis with the 3-year landmark overall survival, 14.6% versus 6.9%. There is a tail on this curve, as we always talk, and there are patients that seem to benefit, and they benefit for a prolonged period of time. The response rates are 26.7%. So still, about a quarter of patients with response. And obviously, improvement in median overall survival, but we still have, I would say, a lot of work to do. And I think we would all agree that we don't want to rest on our laurels here. But important because, of course, it was a major paradigm shift in terms of how we treat newly diagnosed patients with the addition of immunotherapy to gemcitabine and cisplatin's backbone. This was the primary results with the progression-free survival, with again an improvement in median PFS from 5.7 months to 7.2 months. And I think, I don't know how everybody on this call felt, but I was admittedly, I think, a little bit cynical in the beginning because I thought, well, this was a study that enrolled very much in Asia and with a predominance of Asian race. And I will say that for those of us who have been using this regimen over time now, the initial subgroup analysis that looked at multiple subgroups, including primary tumor location, race, region of the world, and PD-L1 expression, it was kind of consistent that we don't really need to select patients to consider the addition of durvalumab. And importantly, when we think about mutations, in this disease with biliary tract cancers, we always wonder whether or not there is something that responds more or less based on the genomic profile. And again, to the credit of the TOPAZ investigators, they looked at what they called the biomarker evaluable population. And that biomarker evaluable population, importantly, they demonstrated that the genomic alterations that we typically think about in this disease were in fact the ones that were seen, including things like IDH1 and ERBB2, and of course things like KRAS. But again, similarly, while these were very small numbers and nothing could really be powered to be definitive, it did not seem that in patients with clinically actionable and prevalent genomic alterations, that there was really any reason to select based on mutation status. That in fact, the addition of durvalumab to gemcitabine and cisplatin demonstrated a similar overall survival benefit. And importantly, and I think we all know this, the addition of durvalumab did not lead to anything catastrophic or worrisome in terms of adverse events with patients. And for those of us who've been using gemcitabine and cisplatin, we really had gotten comfortable with giving the GemCis. And so the addition of durvalumab and what it could do in terms of adverse events was a really important question. And what was very reassuring to see is that treatment-related adverse events in terms of grade 3, 4 were not statistically significantly different with the addition of durvalumab between the two arms. And in fact, there was really not any significant difference in terms of patients discontinuing treatment or God forbid, death with the addition of durvalumab. And what I think, again, is really important, and I think those of us who treat our patients with these regimens really wanted to see and was important for the TOPAZ investigators to look at, was quality of life. These are patients who get gemcitabine and cisplatin, so a platinum chemotherapy for 6 months, and then continue on durvalumab. And again, in that tail on that curve, if patients are continuing, they're continuing on it for some time. And so again, as a kudos to the investigators, they did look at quality of life, and they specifically looked at deterioration of global health status. And really, there did not seem to be any detrimental effect on patient-reported outcomes with the addition of durvalumab. So this was a regimen that improved all the efficacy endpoints that we look for. The safety profile was really favorable, and importantly, quality of life was relatively preserved. Now, not to be forgotten, we have another option for frontline patients, and that's based on the Keynote 966 study. This was a large randomized global study again. This time it was gemcitabine cisplatin with pembrolizumab. This was a larger study, about 1,000 patients or so. And this time the maintenance arm, just as a reminder, after 6 months, the gemcitabine is continued and the cisplatin is discontinued. So the maintenance arm is gemcitabine with pembrolizumab versus in TOPAZ, where it's just the durvalumab alone. And then this was the survival data in terms of Keynote 966. To me, what was really kind of reassuring here, on the left was the initial presentation, on the right was an updated presentation. And again, what I think was reassuring was, number one, we see a separation of the curves with a 36 landmark OS of 13%. But I think, again, what we see is GemCis Pembro median overall survival 12.7 months. So numbers are playing out very similarly between Keynote 966 and TOPAZ, which I think is just proof of principle that chemotherapy with gemcitabine and cisplatin with the addition of immunotherapy really kind of became our primary go-to in newly diagnosed patients. Now, I just want to level set with what some of our other quote unquote standards are before we dive into some of the progress. I'm sure everyone is familiar with the ABC06 study, which was looking at second-line patients, so patients after gemcitabine and cisplatin, randomized to receive FOLFOX versus active symptom control. And again, technically positive study, median overall survival was improved with a hazard ratio of 0.69. But to me, leaves a lot to be desired. Improvement was from 5.3 months to 6.2 months. And we know that we can and should be striving to do better in these patients, but we also know that after frontline therapy, there are a number of patients who, as you can see with that kind of steep drop in the Kaplan-Meier curve, who have difficulty going on to second line and beyond therapies. And then there was the kind of looking at liposomal irinotecan combinations, and that included the NIFTY study. This was again after GemCis, patients who received 5-FU leucovorin with liposomal irinotecan versus 5-FU leucovorin. And this was the initial study of NIFTY that looked at with an OS of 8.6 months. But then we also saw the Naliric study, which looked at a same combination. And here, this was not, I would say, as exciting, with a median overall survival of Naliri 5-FU at 6.9 months versus 5-FU of 8.2 months. So needless to say, in the second line setting, 5-FU based regimens absolutely an option, but I think there's a lot of work to do in that space. So let's talk about what's new. The organizers asked me to kind of especially update on things that we have seen in recent times. And so I know I don't need to talk too much to this group about this, but this was really, I'll be honest, was really exciting and a proud moment for me during ASCO, this year, and being able to see some progress coming out, some important data in the biliary tract cancer space coming out of India. And so this was the Accelerate study, which looked at patients who had resected gallbladder cancer with a PT2 or N positive disease and an R0 or an R1 resection. And these patients were randomized to receive what was the quote unquote control arm, which was chemotherapy alone with modified GemOx or gemcitabine and cisplatin, versus 3 cycles of chemotherapy followed by chemoradiation. And that was concurrent with oral capecitabine, and then finishing out 2 to 3 cycles of the chemotherapy. So kind of a chemoradiation sandwich approach. And this was a really great endeavor. I think it's an important question because when we lump, those of us who talk about this all the time, we know that lumping gallbladder cancer, intrahepatic and extrahepatic cholangiocarcinomas into one large adjuvant or, perioperative study is probably not the best approach for us to really understand how to best treat these patients and how to obtain the best possible outcomes. And so kudos to the investigators for looking at this specific patient population. The study unfortunately closed early due to slow accrual, but the data that was reported out, this was the primary endpoint. This was recurrence-free survival, and you can see that the quote-unquote control arm, the arm with chemotherapy, actually did probably better than one would have expected. And then arm 2 is there in red. And similarly for overall survival, similar results and outcomes were seen. And so the conclusions were, that there was really no improvement by adding chemoradiation to chemotherapy compared to the chemotherapy alone. But, I agree with the author's caveats that accrual could not be completed. And so, a larger trial may be a little bit more informative. This is a very high-risk patient population, as we all know, with T2 or N1 or potentially R1 resections. And so, again, I really think the work that's happening in India to address some of these questions, given the incidence of things like gallbladder cancer, is really is an important work, and I really hope is continuing to move forward. Some of the other kind of exciting things that we know we will see data on, but we at least know from initial report that things that we're looking forward to, I would say, is the Companion 002 study. So this is a study that is looking at tovecimig, which is a DLL4 VEGF-A bispecific antibody. And so we're looking in this study in patients who are on second-line treatment, so have had one prior systemic chemotherapy regimen. And they were randomized 2-to-1 to a combination of this bispecific with paclitaxel versus paclitaxel alone. 168 patients were enrolled, and crossover was allowed. Primary endpoint for this study is overall response rate by independent central review, with obviously key secondary endpoints that include PFS and overall survival. And all we know so far is a press release that has come out a couple months ago that says that this is looking to be a positive study with an overall response rate of 19% in the experimental arm of tovecimig and paclitaxel versus 3% with paclitaxel alone. And here you can see what we know so far. The secondary endpoint analyses are expected to be done kind of, actually, we're probably in that zone right now. I think they're in a data lock right now. So hopefully by the end of the year, we'll be able to see more PFS and OS data and such. And importantly, again, so far, nothing concerning from a safety profile. So this is exciting. You know, I think seeing the entire mesh of data together will be important. But this is, to me at least, important because we're looking at second line, where I said, you know, the bar is already pretty low, unfortunately, and all comers. You know, we have a plethora of options, thankfully, for patients with specific molecular alterations. But, you know, the ability for patients to have options when they do not have a biomarker that is easily targetable or readily available in terms of FDA approved or drugs or clinical trials, I think this will hopefully help us understand what that landscape could look like. So now moving into the space that I think, you know, we have really seen tremendous progress in. You know, I know I don't need to belabor the point that in biliary tract cancers, we have learned over time that anatomic distinctions are important. But in addition, gallbladder cancer, intrahepatic cholangiocarcinomas, and extrahepatic cholangiocarcinomas are molecularly distinct, and they are very complex from a genomic profiling perspective. And, you know, I really will say I was a junior faculty under Dr. Javle when at MD Anderson, when the onslaught of really understanding what next generation sequencing could do and how we could really start to better understand these biliary tract malignancies and then potentially develop therapeutic targets for them. It's Dr. Javle and a number of investigators who really led this charge. And it was really wonderful to be part of, to be able to witness how we could take what was thought of as a rare disease, an orphan disease that pharma was just not interested in, and then really identify potentially targetable alterations for which drug development could be focused around, and then demonstrate and prove that in fact there are enough patients to be able to systematically go through clinical trials and offer drugs to patients. So I'll start obviously with some of the older trials, but then hopefully move into some of the spaces that are developing in each of them. IDH1, as we know, is seen primarily in intrahepatic cholangiocarcinomas in about 10 to 15% or so. The CLARITY study was the pivotal study that was a large randomized global phase 3 trial that looked at an oral IDH1 inhibitor, ivosidenib, compared to placebo at that time in patients who had IDH1 mutated cholangiocarcinoma and who had progressed on 1 or 2 prior lines of therapy. At that time, we did not have FOLFOX as the benchmark, and so placebo was the control arm, and so crossover was allowed. Primary endpoint, therefore, was set at progression-free survival. And this is the primary endpoint with a numerical improvement in median progression-free survival from 1.4 months to 2.7 months, with a strikingly positive hazard ratio of 0.37 and a disease control rate of 53%. This is a relatively cytostatic drug. That partial response rate was only 2%. But, you know, for those of us who have used this drug, very well tolerated, easy to give our patients, and, you know, absolutely a clinical benefit that is derived from the use of ivosidenib, and is now FDA approved and available. So obviously the questions always become when we have new drugs, how can we build on them? What can we do? And so, you know, as a testament to collaborations in this space, there's been a lot of really interesting preclinical data that suggests that that you can combine and in fact see synergistic effects when you use ivosidenib in combination with GemCis in IDH1 like PDX models and such. And there was in fact superior tumor growth inhibition versus GemCis alone. And so some of that data, as well as some really interesting data that looked at combining immunotherapy as well with this inhibitor, has led to some important questions in the combination space. So this was a study that looked at gemcis with ivosidenib. And this was in treatment naive, newly diagnosed patients, 13 patients. And the data presented here basically showed that with a median follow-up of 4.1 months, the overall response rate was 46%. And at that time, the median PFS was not evaluable. And importantly, combining ivosidenib, which is, like I said, a relatively well-tolerated drug, was safe to add to the gemcis backbone. And so then, of course, the landscape changed, and we now use gemcis durvalumab or gemcis pembrolizumab as a frontline regimen. And so there's now a phase 1b/2 study that is going to be looking at ivosidenib with the gemcis durva combination, and a consideration of maintenance ivo plus durvalumab, with then subsequent expansion and extension, and all of that will kind of be based in a stepwise progression. And so, what we're looking at now is an important question of, once we confirm safety, given ivosidenib being well tolerated, and then adding it to gemcis durva, then to really understand whether what we see in the preclinical space, is there something truly synergistic that we see when in IDH mutated cholangiocarcinomas when we add ivosidenib to the gemcis durva backbone. So, I think a lot of us are looking at this with great interest. Moving into FGFR2, this story has been ongoing for a very long time. FGFR2 fusions are, seen in about maybe up to 10%. It's a rare finding, but FGFR2 fusions in particular are what were looked at first. Fusions or gene rearrangements. And the first kid on the block in terms of publications coming out was pemigatinib. Pemigatinib is an oral FGFR inhibitor. And the FIGHT 202 study was a multi-cohort study that included an FGFR2 fusion or gene rearrangement positive cholangiocarcinoma cohort. And this was the updated results that came out for that cholangiocarcinoma cohort. And as you can see, with a median follow-up of pretty much 4 years, overall response rate with pemigatinib was 37%, with median duration of response of 9.1 months, PFS of 7 months, and a median OS of 17.5 months. So, this drug was proof of principle that we needed to identify FGFR2 fusion patients and offer them FGFR inhibitors that we can target this pathway. And I think what was really refreshing to see when the updated results came out is the overall response rate holds. It's still about the same as when we saw the initial data, but that duration of response, when we think about the other options in the second line and beyond space, I think it's really impressive to see what we see with response as well as duration. Similarly, futibatinib is another oral FGFR inhibitor. This is the data that was presented or that was published in the New England Journal by Dr. Goyal and colleagues that looked at 103 patients with FGFR2 fusions. At the time of publication, the median follow-up was 17.1 months, and the overall response rate with futibatinib, which is slightly different in terms of the type of FGFR inhibitor it is, but still, relevant in patients with FGFR fusions regardless. The overall response rate was 43%, and again, a median duration of response of over 9 months. PFS was 9 months, and median OS was 21.7 months. So, those are the two drugs that then became available to our patients for with FGFR fusion or gene rearrangement positive cholangiocarcinomas. And importantly, these are drugs that we have learned. There's been a learning curve in terms of management of the toxicities. The hyperphosphatemia, which is a class effect that we know from FGFR inhibitors, was, I would say, kind of an important one that was followed very carefully in all of these protocols, with clear management of what to do in terms of using phosphate binders and low phos diets and things. But to be honest, the clinical relevance of hyperphosphatemia, thankfully, I think probably because we knew how to manage it, was relatively low. It's actually some of the other things, the cutaneous toxicities, for those of us who take care of these patients. Dr. Javle, I know for a fact, he and I have talked about some of these things in terms of the dry skin, discolored nails, the dry eyes. There's serious toxicities like retinal toxicity. Those are relatively rare, but it's the ones, these patients stay on these drugs for a while. And as a result, some of these things are really, difficult to manage for our patients, with the dry eyes, the nail changes, et cetera. But you can see here that they're very similar regardless of the type of drug that we use. Now, that was I would say, the old generation of FGFR inhibitors. We have a whole new bevy, thankfully, of FGFR inhibitors that are what I would call newer generation drugs. And this, for instance, is an example of some progress that we're making. This is tinagotinib's data that Dr. Javle presented at ASCO GI in 2024. And again, this is an important drug. This is a drug that is, like I said, a newer generation and had shown some potential activity in number one, FGFR2 fusions, but also in other FGFR alterations, as well as in potential for patients who have acquired resistance to prior FGFR inhibitors. And what you can see here is the FGFR altered cholangiocarcinoma patient overall response rate was 26.3%. And then when you specifically look at acquired resistance patients who have had prior FGFR inhibitors, which was admittedly 10 patients, but overall response rate here was 40%. And then when looking at FGFR2 mutations, FGFR3 amplification and rearrangements, et cetera, 13 patients, again, overall response rate of 30.8%. So this drug, I think, to us is really exciting. And, again, testament to Dr. Javle's just kind of continued efforts in this space, because we have patients now who have received pemigatinib and futibatinib, and what do we do with them? As well as, I think a number of us get those next generation sequencing reports that have things outside of FGFR2 fusions. And so having potential for having drugs available to these patients is really exciting. So this data, of course, makes us want to ask the larger question. So this is the first 308 phase 3 study of tinagotinib. And here you can see the plan is for 200 patients for a 2 to 2 to 1 ratio of tinagotinib at 10 milligrams versus 8 milligrams, and then physician's choice for the part A. And then there's kind of a part B that looks at the selected dose, if you will, with 100 patients planned there. And there, the primary endpoint will be progression-free survival. And so, this study, I think, will give us better understandings around what tinagotinib can truly do. But again, is exciting to know that there may be another kind of different mechanism and a potentially different drug available to our patients. HER2 is another area of excitement. HER2 amplifications we see, I would say, skewed more towards extrahepatic cholangiocarcinomas and gallbladder cancers, about 10 to 15, up to even 20% sometimes. But we can see them across biliary tract malignancies. And this was the Horizon BTC-01 study, which was the phase 2b study that looked at zanidatamab, which is a monoclonal antibody bispecific that targets HER2. And these were patients who had HER2 amplified biliary tract cancers. And there was the primary cohort, which was IHC 2 plus or 3 plus, HER2 positive patients, and then a cohort 2 that looked at IHC 0 or 1 plus. And they received single agent zanidatamab, and primary endpoint was overall response rate. This was the updated data from the Horizon BTC study. This was 80 patients with HER2 amplified BTC, specifically looking at that cohort 1. And median follow-up was 21.9 months at the time of this report, with an overall response rate of these refractory patients of 41.3%, and a median duration of response of 14.9 months. So again, really impressive durable responses with a median OS of 15.5 months. And so this drug received accelerated approval at the end of last year. Now, obviously, there is an ongoing phase 3 study, the confirmatory study, if you will. This is patients with 286 patients, HER2 positive, that are being combined, looking at zanidatamab with GemCis plus or minus a PD-L1 inhibitor. And so you can see the schema for this study. This study is open and enrolling now globally. And then in terms of HER2, the other big splash, I guess, is a tumor agnostic approval that came out of the Destiny Pan-Tumor02 study. This is looking at trastuzumab deruxtecan, which of course has gotten a lot of press in terms of its activity in breast cancer, as well as across a number of other malignancies. This is the antibody drug conjugate that specifically looks at HER2. And in this pan-tumor study, you can see that there was a biliary tract cohort, that's the cohort 1. And that study, the efficacy across 41 patients is listed, but when you specifically look at the HER2 IHC 3+, which was 16 patients total, there the median duration of response was 8.6 months. The OS was 12.4 months. And there was no surprising safety signals. And so this drug is now also available again in a tumor agnostic manner for patients with IHC 3+ HER2 amplifications. I'm just going to briefly touch on KRAS. This is obviously a rapidly evolving study, and the Crystal-1 study is just one example of the proof of principle that we can now target KRAS, and which I think is going to create a sea change in the management of GI cancers. I think it's safe to say. This is specifically targeting KRAS G12C, which admittedly is very rare in pancreas cancer, biliary cancers, et cetera. It's rare across most GI malignancies, in fact. But in this study that looked at KRAS G12C mutated GI cancers, there was some pancreas and some biliary cancers included. Overall response rate was 41%, but the response was 50% in biliary cancers, with a duration of response across all GI malignancies of 8 months. So again, just kind of proof that we can, in fact, target KRAS, specifically in biliary cancers. But, I think obviously everyone's eyes are on some of these newer RAS inhibitors coming out of Revolution Medicine, for instance, the daraxonrasib, for instance, which is an exciting drug and being studied very comprehensively, I would say, in pancreas cancer. Unfortunately, we don't really yet know what these are going to do in the world of biliary tract cancers. These are really promising drugs. This is just a snapshot of some of the phase 1 data that we've seen in this specific drug for patients with pancreas cancer. And there's an ongoing phase 3 trial right now with daraxonrasib with pancreas cancer. And I think, it's going to be really important. And again, foundations play a key role in terms of patient advocacy and advocating to the companies in terms of making sure that we have some access to looking at these drugs in a really systematic trial format for biliary tract cancer patients. KRAS is a very relevant, as we all know, mutation for biliary tract patients, and so again, as a testament to a number of thought leaders across the world, we know that there are studies in development here. BRAF V600E mutations we know are rare, 3 to 5% of biliary tract cancers, primarily intrahepatic cholangiocarcinomas. This is just the data that was published many years ago, but that led to the tumor agnostic approval for dabrafenib and trametinib, combination of BRAF and MEK inhibitors, looking specifically in the biliary tract cancer cohort. And in that cohort, the overall response rate was 47% when we combined dabrafenib and trametinib in patients with BRAF V600E biliary tract cancer. So just to kind of, that was a brief glimpse into some of the things that we have and that are coming down the pike. But we have, as you can see, based on either tumor agnostic and or cholangiocarcinoma specific or biliary tract cancer specific approvals, we have a number of targeted therapies that are available now to our patients. That includes, the FGFRs, IDH, HER2s, but also very rarely seen, but tumor agnostic approvals and things like NTRK fusions, RET fusions, et cetera. NRG1 fusions. Apparently, there's some interesting efficacy in patients with biliary tract cancers that have NRG1 fusions. But there's also a number of new targets that are emerging. And, I think it'll be important for us as we talk through what we can and can't do together as a global community, how we can really bring some of these other more rare alterations, how we can bring these trials together in a global fashion so that we can really make sure that we have space and access to these drugs across the world. So just to kind of briefly summarize, this is our current treatment landscape. We have our GemCis plus immunotherapy right from the get-go. And then we have all-comer, no targetable alteration drugs available, potentially with the addition of Companion 002 once we see that data. And then we have the targetable alteration space, including what's currently available. But as you can see, there's a lot happening. This is just such a wonderful space to be in right now. there's a number of second generation, if you will, drugs in the FGFR and IDH and HER2 spaces. I already talked about KRAS, but as I mentioned, there's also other targets, and that includes things like MTAP loss. Dr. Javle and I and others have been talking about this space for a while. Claudin 18.2, which is a very relevant target and already has led to some FDA approvals in other diseases. Homologous recombination targets, TROP2, HER3, and then of course cell therapy, where there's a lot of really great interest looking at potential targets such as Claudin 18.2 or MUC. So lots of trials that are ongoing that I think just kind of summarize that there is now momentum and excitement in this space to really better understand how we can move the needle in biliary tract cancers. But I think the question is how do we be nimble? I just want to highlight two what I think are just wonderful initiatives. One is the Saphir ABC10 study, which I'm not going to go into the specifics, but again is out of Europe, based out of the UK, just like the ABC group does. And again, this is the idea of bringing in patients at the time of diagnosis, doing molecular profiling, having a molecular tumor board, and then after some standard of care, what are some potential maintenance options based on if they have an actionable alteration? And so they get, they go through this very systematic process to ensure that everyone's getting molecular profiling, which I think we can all agree needs to be happening. And then there's an important molecular tumor board conversation around, do we keep them on standard of care or do we move them into maintenance that specifically goes after a target? And similarly, again, through Dr. Javli and others' leadership, there's similar efforts in looking at how we can do platform studies and kind of master protocols that would allow for more nimble discussion and understanding of new targets. And so this is the Arbor BTC schema. This is, again, an effort that's happening in the US right now to better understand what we can do with patients as we get genetic testing done, and if they have an aberration, how we can test new drugs in a quick fashion to get kind of quick understandings and readouts and proof of concept to then be able to move more stepwise towards registration and drug availability. So in summary, I think targeted therapy is at the forefront of managing biliary tract cancers, and multiple drugs are approved in the refractory setting. But right now, we have GemCis and immunotherapy in the frontline setting. We have established targets such as IDH and FGFR2 with newer generation FGFR inhibitors, I think really showing progress in the acquired resistance population and with the ability to have those drugs hopefully in hand soon. HER2 has a number of drugs available with a variety of mechanisms of actions, and we've got KRAS inhibitors that we've shown proof of principle, and we need slots in these pan-RAS trials. Their tumor agnostic approvals have been pivotal to allowing for us to have access to rarer targets, but patients who really need these therapies, and other targets are emerging, including opening doors to novel drugs and to cell therapies. And novel platforms for trial design, I think, have been and will continue to be key to advancing the field. I can't tell you. I mean, whenever I give these talks, I get goosebumps because I really think this disease remains the model for precision medicine. And it is a testament to the investigators across the world that so many drugs have moved forward so quickly. And like I said, it's really, really just humbling to be a small part of it because watching it has been, it keeps me going. It continues to give me breath. So with that, I will stop and happy to talk through any questions.
Thank you, Rachna. That was such a phenomenal talk. And everything that was covered is what we look up to, because we do practice frontline application. Topaz, maybe 1% of India is able to get durvalumab, is able to see the ideal treatment, frontline treatment option as per the standard of care. But the truth also remains that we are here in India short of clinical trials, and access to clinical trials, of course, brings in novel therapeutics. As we get some comments in the chat box, I will begin by asking a few questions. First of all, this was a stellar talk. I have taken notes, and it was great to catch up as to what is standard of care and what is novel and what is on the horizon to watch out for. One of the challenges that we have had in India is whenever a patient gets diagnosed and you're starting frontline therapy for someone, you wonder whether it's still standard of care or have you moved on in frontline setting? Are there trials to watch out for? There are patients who will ask you this question. Is Gemcis Durva the main thing happening in the US right now? Or if you got a patient frontline with cholangiocarcinoma, advanced cholangiocarcinoma, would you enroll them to a particular study? And we are interested in knowing which one.
Yeah, and Dr. Javle, I'm sure, has his opinion here. I mean, I think I will say, and this is going to sound like a very stereotypical answer, but if we have a clinical trial available, I most certainly offer our patients a clinical trial first. I mean, at the end of the day, I always tell patients, we have Topaz, we have durvalumab now because 685 patients went on a clinical trial, right? So my hope is that if we have a clinical trial that I think is smart enough that I've thought through the process to open it, then yes, I absolutely offer them a clinical trial. Do I necessarily think that I have an all-comer trial open? Like, for instance, I don't have an all-comer trial open right now. There's a lot, as you saw, that I presented that are in frontline setting for very biomarker-specific patients. And of course, there's the complexity around having that biomarker data available in time, but most of these studies are now written to allow for starting the chemo and or immunotherapy before putting them on these studies. But there's a Destiny BTC study that's for patients with HER2. There's the tinagotinib study. There's the Horizon study. I mean, so those types of studies, I think, if I know that those patients have those targets, those are absolutely the types of studies I try to put them on, because we need to answer those questions, and that's the other part, that from the company perspective, there is the regulatory need, but I think for all of us, we really want to know. We want to know if these patients do benefit in newly diagnosed HER2 amplified BTC. Do they benefit from going on Gemcis, Durva with, trastuzumab deruxtecan? Like, I think that's an important question that we don't really know. Is there a synergy, or should we be doing this in subsequent lines of therapy? Dr. Javle, I don't know if you have anything else to say there.
No, I think you said it very well. And I want to say, Sevanti, that, for instance, we have learned so much from what you folks are doing in India. The gallbladder study with Rina engineer Vikas Oswal, where they gave chemoradiation before surgery. So we are trying to do a study where induction chemo, immuno, followed by radiation therapy. This is the kind of study that can be done in India, like by you and like Prasad or somebody, where you give induction chemo, half the people get radiation, half the people do not, or answer the multi-modality question. But there are, like Rachna said, we have one of the agents she described earlier, like that is from the companion O2 study, CTX009. We have a all-comer study for that with Gemcis and Durva that one of my juniors is running, and various other targeted therapies like ivosidenib first line, HER2 first line. If we find those patients, that would be the way to go, I think.
Yeah, great. No, I do see a lot of effort happening here across many centers in India. We have a large cholangio gallbladder population, and local oncology centers, leaders are emerging with questions that are very, very relevant. And kudos to them, because these are all very, very pivotal local questions which need to be answered and can be answered in simple study designs. I have a question from Dr. Saumil. He's one of our esteemed panelists for this evening, and he's interested in knowing, and Saumil, you can ask your own question as well. He's interested in knowing what is the neoadjuvant for a borderline patient. Do you approach with a neoadjuvant regimen to get a borderline unresectable patient into resectability?
Yeah, this is such an important question, and frankly, we probably could use help from our colleagues in India to be able to systematically answer these questions.
Yeah.
Because, I would say us in the US, we have a tendency to chase our tails on these types of really low-hanging fruit, but clinically relevant questions. Um, I will say the short answer is I think most of us nowadays will take something and look to see if there's a potential for conversion. Whatever we don't have a clear definition like we have in pancreas cancer of what borderline resectable really means. But, in a multidisciplinary tumor board conversation, there are absolutely things that are happening, and typically, when we work with our surgeons, we will talk through different approaches to try to quote unquote downstage or convert somebody to resectability. Dr. Javle mentioned SWOG 1815. I didn't talk about it because it was, technically a negative study. But I will say that GAP, which is what I lovingly call it, gemcitabine, is still, I think, a very relevant drug in that space. The NEO-GAP study demonstrated the feasibility of giving preoperative gemcitabine, and I know our Indian colleagues, our Korean colleagues, a number of other countries have looked at that triplet regimen in that space. And I will say I use it. I use it in patients that I think are borderline resectable and have continue to see results.
Yeah, I see this regimen offered even from Tata Memorial Hospital all the time. I have patients who come to me second, third line, and they have been treated with this combination, and some of them have been responders, durable responses seen. There is a question regarding tinnigotinib and whether the trial is something that you look up to, you enroll frequently, and how is the trial doing? I don't know whether this is a confidential question.
That's a Dr. Javle question. I'll let him answer.
Okay.
So yes, the trial is is actually doing well because it's sort of a third line trial after chemo, one FGFR, and that stopped working, then the third line trial. And that trial compares it to either FOLFOX or FOLFIRI. That's the first 308 trial. Of course, the sponsor was ambitious, expecting the trial will be done by October. I'm not sure about that, but it looks like it will be completed. And of course, the results we will wait and see. I think I'm just excited that we will have a phase 3 FGFR study that will actually be completed. And so we'll learn a lot about how to treat these patients afterwards. And by the way, it has a compassionate use program, which is available to patients even in India. So I've campaigned for that. And if you have a patient who you feel may qualify, it will require some paperwork, but it will be feasible to get it.
Thank you. I have a couple of other questions here. Dr. Prashant is checking in if we could use an IDH1 inhibitor in frontline therapy, or how early is it possible for us to utilize an IDH1 inhibitor? And
Yeah,
then
so technically the indication right now is in second and third line for ivosidenib. But as I mentioned, if you have the ability to put patients or get patients on these trials, there is a trial right now that's looking at the gemcis durva with ivosidenib combination. And so, I hopefully will have the answer to that question soon. Again, this is what I was saying, that if in the frontline setting, if we know they have an IDH positive, if they have an IDH mutation, I try to get these patients on these types of trials. So to Dr. Javle's point, so we can actually answer these questions, but that would be the answer right now.
great. And then we have a question from Dr. Preetam Kataria, who's a very prominent medical oncologist in our team at Reliance Foundation Hospital. He asks if there's a patient with HER2 positive disease, would you consider the TOPAZ regimen, or would you combine a HER2 off of a trial? I mean, I think on a trial would, of course, be HER2 directed, but off of a study, would you add a HER2? What would you do in your practice?
Yeah, I think, in patients with HER2 amplification right now, I would give them their primary approach would be to give them single agent, one of the drugs. That being said, the reason that they're looking at these frontline studies, aside from a confirmatory perspective, but the gastric cancer is a perfect example of demonstrating synergy between immunotherapy and anti-HER2 targeting approaches, right? So one would think that there should be potential for additional efficacy or impressive efficacy outcomes when we combine anti-HER2 approaches with durvalumab or with immunotherapy as a general approach. And so again, I think that's why these studies are going to be important studies, the Horizon Frontline study, as well as the Destiny BTC study. The Destiny BTC study actually includes the Rilvi, right? Right, Milind? It's rilvegostomig too. So it's an
I totally
interesting.
agree with Dr. Shroff, Pritam. So yes, I mean, I've learned that watching how Semanti treats her patients. If you wait for every trial to come out, it's not possible in India. So if you have something simple like trastuzumab in combination with Gemcis and Durva, I think it's reasonable. I mean, we know that there's combination data in gastric cancer, right, with pembrolizumab and HER2 blockade, and those tumors are more immunogenic. So I think that, I mean, in fact, I was doing that before we had these trials available. So it's reasonable. I think it's, for us, we have the luxury of saying in America, let's put them on a trial. Well, you don't. So I think if the hypothesis is reasonable and the toxicity is manageable, um, I would go ahead.
Thank you. That was very helpful. I'll ask the last question and then we move over to the case presentations. FGFR inhibition. So we have, of course, we don't have these drugs. We've imported, we've given. We find them very difficult to deliver because of the toxicity. The newer FGFR inhibitor, are they much better tolerated, or are we still struggling with toxicity similarly as the earlier generations?
So I would say they seem to be, but Milind, I mean, you probably could give a little more nuanced answer, but I would say overall, because they seem to have a little bit more specificity, I think they seem to be. But Dr. Chavle, what do you think?
So I agree with Rachna, but like all of these drugs, Semanti, there's like a learning curve. So when you treat enough of them, then you know how to manage it. I mean, we deliver really toxic chemotherapy. So relative to that, this is much manageable. And I think, just reach out to people who have used them because Rachna and I managed to deliver them sometimes for a year or longer and just find ways to mitigate those toxicities with hyperphosphatemia, management, the skin, etc.
Right. I think that's a great answer, because I feel a lot of these toxicities with many difficult to deliver drugs we have been able to master with the help of multidisciplinary support. And here also there is a learning curve, and as we warm up to bringing more drugs in to address pathways, we must be aware and educate each other and seek help and guidance and manage these toxicities. With that, a huge thank you to Dr. Shroff for such a stellar talk and for all covering the bases, the entire bases, and answering all the detailed questions. With this, we will move over to the two case presentations. We will start with Dr. Akash Tiwari, who is young budding medical oncologist at Jupiter Hospital in Indore. Akash, thank you so much for presenting this case. Dr. Akash Tiwari is presenting a case in gallbladder cancer, reflecting management of advanced gallbladder cancer in the current era in India. Please take it over from here, Akash, and then we will bring in our panelists. I just want to mention them quickly. We have Dr. Prasad Dandekar, who is the head of radiation oncology at Sir H.N. Reliance Foundation Hospital. We have Dr. Saumil Vyas, who is a senior surgical oncologist, biliary tract and liver surgeries, gallbladder surgeries are his forte and area of expertise. Dr. Somil Vyas is here today as a panelist. We have Dr. Dhaval Thakkar, who's a young rising star in interventional radiology. And we have Dr. Prashant Kumar, who is a molecular oncologist, molecular scientist. He is from Datar Genetics, and he's a celebrated scientist. He's also here with us today for both the cases as a panelist. So over to you, Akash, for your case. Thank you.
So, thank you, ma'am. Thanks for the opportunity. We know that amongst the biliary tract tumors, gallbladder cancer forms a bulk of the majority, especially in the northern India and the Gangetic belt. So, what we have done here is that we have a small case in an advanced gallbladder cancer. What we have decided is that we'll run through the case. There's certain questions that we formulated. We'll run through the questions as we go by and then have the panelists answer it at the end of the case. So that's how we were thinking to do it. So this is a 39-year-old female who presented to us with pain in abdomen for 20 days, itching all over the body for 15 days, a yellow colored urine for 7 days. She had no comorbidities, was ECOG PS1, had icterus, excoriation marks all over the body, and a tenderness under the right hypochondrium. In August 24, she underwent a USG abdomen, which showed a gallbladder mass with portal lymphadenopathy, but there was no IHBRD at this point. The gallbladder was distended. It revealed endoluminal soft tissue mass in the body and the neck. There was a large portal lymph node, and there was loss of fat plane with both head of pancreas as well as the second part of the duodenum. So it's essentially an unresectable CA gallbladder. From the date of her diagnosis and in the first imaging to the time she presented with us, she had developed jaundice. So we see that on 14th, her bilirubin was 2.53, with direct bilirubin composition being 1.83, which rose to 10.7 to 6.61 direct composition over a period of time. So this lady underwent PTBD and a biopsy from this site. The biopsy revealed adenocarcinoma, which was moderately differentiated. We, from here on, got a HER2 testing, which was negative, and MSI testing. Once we have finished with the presentation, we'll have this word on PTBD versus ERCP stenting from the interventional radiology colleagues. What is the role of HER2 testing upfront and the directed therapy? I think we had a brief discussion on it earlier. And as we did only HER2 and MSI, do people do PDL1 testing also? And the role of PDL1 based choosing of the IOs. So we got a PET CT done for this lady. There was a similar mass in the neck and the body. There was another mass which was infiltrating the duodenum, another mass in the fundal region that was infiltrating the hepatic flexure with a large portocaval lymph nodes. So we have a picture of those, and we see that it is a fairly extensive disease. This lady subsequently received 6 cycles of GEM-CIS with Durva every 3 weeks. And she tolerated it well with some anemia, neutropenia, and admission for febrile neutropenia and mild IO-related toxicity. Once we are done, we'd also like to discuss if people use other options of GEM-OX from the data of Professor Atul Sharma and other IO options in this setting, and how do they compare with each other. So on the end of the therapy in November 2024, we had a significant reduction in terms of both size and FDG activity, both in the primary mass and in the lymph nodal mass. So the pictures above were the current scan at that time, and the below were the comparator studies at the baseline, and we show that we have had a wonderful response. We continued this lady on durvalumab, 1500 milligram every 4 weeks. She was regular with her treatment, no significant toxicity, and we got a PET CT done somewhere in April 25. And at this time, she had progression, especially with the increase in size of the lymph node and the increase in size of the primary gallbladder disease, which was suggestive of progression. And we can see it in the images too, with the increase in both the primary as well as the lymph nodal disease site over a period of time. This time we got a biopsy done again with an NGS, but we could not find, unfortunately, a targetable mutation. And we started this lady on Folfox. She has presently completed 4 cycles of Folfox, no significant toxicity, and she is due for reassessment. Thanks. So,
Great case, Akash. I think questions that you put up were also very, very well placed. I think these are the challenges. So I actually have a question for you before we move over to the panelists. And we have Dr. Javle and Dr. Shroff is also there for the case discussions, Priyanka.
Yes, she is there. She's
Okay.
available. Yeah.
Okay, thank you. So before I move over to the panelists, just trying to understand. So in the current times, the patient has had a recurrence. I saw 2 areas of recurrence in the slide. Is there a like a oligo progression or oligo recurrence? Recurrence would be the wrong word to use, but oligo progression.
so ma'am, yes, there are 2 sites that were active earlier and had disappeared in the interim scan that have reappeared. Not sure if the LIGO progression concept works in CAGB2. Of course, you and the panelists will help me out with that. But yes, there were only 2 sites of the disease.
Okay, very good. So I will begin by requesting Dr. Dhaval Thakkar to weigh in here on one of the questions that Akash had presented. The patient had come in with a high bilirubin, and the question here Akash had presented was, how do you decide between ERCP versus PTBD, and are there other ways to get the bilirubin down? Is Dhaval here? Well, I guess he's not.
Right.
Hmm?
So can I come in, Sevanti?
Yeah,
Am
yeah.
I?
Yes, please.
Yeah. So I think this has first of all, it's a pleasure to be here this evening. Thank you to Dr. Javle, Dr. Sevanti, Priyanka, of course, for having this continued educational forum. And it's a very nice, interesting case that has come up here, a scenario that we typically face, more or less in lines with the question that I posed in the chat box during Dr. Rachna's talk. So I think how do you get rid of obstructive jaundice and bring the bile back to the bowel in a patient typically with advanced biliary tract cancer? And the general convention has been that if you got a block above the level of the hilum hepatic confluence, then the conventional method has been PTBD. You approach it percutaneously, and if you got a block below the level of the hepatic two ducts confluence, then by definition you got a cholangiocarcinoma, which is at below, and you approach it with an ERCP. The other way also of looking at it is that if you got a type 4 hilar block, which means there is a separation of the ducts, which you would see on MRCP, and the ducts are not communicating, then regardless of where the block is, you do approach it through PTBD alone, because ERCP will not be able to get into that biliary system and cannulate both the ducts. So at the end of the day, I think it's a comprehensive discussion with the radiologist, with the oncologist treating the patient, the medical gastroenterologist, And it also depends on what the expertise, local expertise is available. If you got an expert endoscopist who can navigate across a very tight biliary tree, get into the biliary system and achieve effective decompression, that's ideal, because the advantage with an endoscopic approach is that it's a one procedure approach. You don't have to stage it out. You just get into both the systems. You can get good cytology, and you can really do it at one shot. Especially if you're dealing with a patient who's got an advanced cancer, which is purely palliative, then you prevent the distress of repeated PTBD procedures. The disadvantage is, of course, that if you cannot navigate in and you've gone into the system, you've introduced infection, and then you get caught up into a spiral of recurrent cholangitis and sepsis, which can actually put the patient back in a very significant way, because they don't really come up for getting any effective chemotherapy, systemic therapy, or operations also for a very long time. So I think there is no clear-cut answer. There is no real study which has addressed this or brought out any distinct advantages. But what is important is to have a comprehensive discussion, local availability, local expertise, MRCP, and a comprehensive discussion with the interventional radiologist and your endoscopist. I think that's where
Very
we
well,
should
very well
decide. Yeah,
said, very well described, and thank you for coming in here.
yeah,
Somil, while I have you, I actually
yeah,
had put out a question for you. Both at the time of first response as well as currently. So once they have again reassessed the patient, and because the current presentation was mainly with two lesions, what would be how would a surgeon come in here in
yeah.
terms of a radical surgery? Is there a scope for radical surgery, considering gallbladder tumor is not is not a very radiation sensitive disease? How would you
That's
think, what would be the role of surgery?
Yeah, so I think when the first scan was put up, there was encasement of the main portal vein and hepatic artery, and there have been two publications on the subject which have actually defined a borderline resectable gallbladder cancer, more or less like borderline resectable pancreas cancer, which came out from MD Anderson in 2006 in the first instance. So there was a publication last year in HPB, which was essentially a questionnaire, but they have tried to classify borderline resectable gallbladder cancer and bring out a consensus definition. And this would fit in more or less with that, because you've got encasement, more than 180-degree encasement of the vein as well as the artery. So the right thing to do was go ahead with systemic therapy. There may be a school of thought that you could do a staging laparoscopy at the time of initial diagnosis, because you're going to upstage the disease in at least 25% of the patients. There is a very good paper from William Jenargan from Memorial Sloan Kettering, which has shown that you could upstage almost 25 to 30% of patients on whom you laparoscoped. And then, of course, they then get classified as metastatic, which means that the concept of downstaging them to bring them up to an operation, that question disappears from the equation. Now,
Right.
assuming that this patient is locally advanced, I think at the end of completion of his gemcitabine and durvalumab cisplatin combination, maybe that was a time to look and assess for resectability, which again means that do a laparoscopy and give this patient a trial resection. I think she's 39 years old. The performance status is 1. And if you had a reduction of the nodal mass, then without offering a definitive resection, you're not really going to achieve a cure. So, and even now, I mean, it may not be a wrong time to consider that, given that there has been mild oligoprogression, but she's not clearly metastatic in that sense.
Yes, so that was a great insight. I think the challenge, of course, is always when you have a patient that is either locally advanced or advanced, is to pick up that time where, leap of faith and go in for a radical surgery. Um, I think we need to move over to the next question, which is, we did the biomarker testing, and in this situation, we didn't find anything specifically. But we have Dr. Prashant Kumar in the panel, and I would like to check with him. What do you usually test for when you have a gallbladder patient? What would you suggest that we look out for in a case like this?
So hi everyone. I think that's amazing meeting, and I first thanks Dr. Sevanti, Dr. Milind, and Dr. Shah. I think you have touched base every domain, and not much to be said now. Biomarker is something which is very interesting, and I just would like to say to the audience that when we talk about the biomarker, it is essentially a biomarker which is at the level of diagnostics, prognostics, predictive, and now we are talking about the emerging landscape of biomarker, which is more personalized. I'll just go one by one. As a diagnostics, we use CA19, and I think this is not very sensitivity. There's an issue, but there's a KRAS, there's a P53, there are microRNAs 21155. These are the very good established diagnostic biomarker. Prognostic biomarker, when we talk about the P53, HER2 overexpression, and once we see the P53, it shows that there's an aggressive disease. Similarly, for HER2 overexpression, which correlate with the poor survival, and of course PD-L1 expression, which is again worse prognosis. A predictive biomarker, when we talk HER2 amplification and 10 to 15% GVC, we know that there's HER2 amplifications. There is also TMB high cases where we predict that when TMB is high, it may respond to immunotherapy. Similarly, FGFR2 and IDH1 mutation, which is not very common in case of gallbladder cancer. Emerging biomarker, I would say that liquid biopsy, which we not very routinely use, but I think ctDNA based liquid biopsy biomarker and BRCA1/2, if we get, it may confer the PARP inhibitor could be used. I am more basic researcher, so I always believe that the EMT is one such areas when we found EMT or a stem cell biomarker like CD44 or ALDH1, which also signify the association of drug resistance. And I think we are in the era of personalized medicine. So NGS panel, which could give us an actionable targetable mutation or the CGP. And I think we should foray in the area of multi-omics, where genomics, transcriptomics, proteomics could be integrated to identify any such biomarker, which could be the culmination of all biomarkers at the level of DNA, RNA, and protein.
Thank you. Thank you, Prashant. I have now moving over to Prasad Dandekar. Prasad, any role of radiation in the recurrent setting or even in the first response? Is there any role here that you would think of a consolidative radiation strategy? Prasad, are you there?
Yeah, I'm here. Thanks, Dr. Sevanti, Dr. Javle, and the whole team for inviting. Excellent conversation. Sevanti, before answering that question, let me just speak a little bit about what you mentioned about cholangiocarcinomas not being very radiosensitive. And one of the challenges, we are not able to deliver that dose there. That has been the major challenge to cholangiocarcinomas. And while we may think that the problem is the liver, but liver is actually not the problem. For us, the big problem is the duodenum. And duodenum really limits the amount of dose. The maximum tolerated dose for duodenum is 45 gray. And if I were to give 45 gray, that's I would say a suboptimal dose. So Rina's data, which Dr. Jamia alluded to, the preoperative dose, they have given a good 54 to 60 gray in these patients. And I think that's a very selected cohort of patients. Now, in this patient, one thing I agree with Saumil, whether at the instance of completing the first line chemotherapy, we could have explored the option of surgery. This particular patient, I wouldn't say was a great patient to do the neoadjuvant CTRT because again, the volume would have been too large and it is involving the hepatic flexure and other surrounding areas plus the lymph nodes. So this is not really a great candidate. But I think in other patients, the role of neoadjuvant CTRT is something that we need to talk about and focus on. So this patient would, I think the major thing was that if the surgery is possible, again, post-operatively also giving radiotherapy in this patient would depend entirely on what we get out of histopath and how the structures are post-operatively. But major issue for us has always been the duodenum.
So, excuse me, Prasad. We, as you know, Rachna and I come from an institute where we are big believers in radiation for biliary tract cancer. So, not in the setting of neoadjuvant, but would you consider as a consolidative strategy, is what Sevanti mentioned, because the patient has had two. We don't really have great options, to be honest with you, after Gemcis and Durva and Folfox. And perhaps radiation will not have long-lasting control. But do you see any role here in that setting, in the palliative setting, not sort of in the neoadjuvant setting?
Definitely, palliative radiotherapy for biliary tract cancers is used very often and is quite effective. And again, we can give a reasonable dose there from palliation point of view, so that can be considered. And if this patient would undergo surgery, then and depending on if it is R1 R2, um, I wouldn't imagine this to be R0. We could again consider the option of post-operative radiotherapy, but if surgery is not on table and the patient continues to progress, we can definitely offer radiotherapy as a palliative radiotherapy.
Thank you. And Akash, I wanted to make the point, it's a little difference in practice. Like, I don't know, I'm sure Rachna as well. We don't order those many PET scans. In India, I see primarily patients are followed with PET CT imaging, which we don't use a lot, actually. I have to say, probably order like 10 PET scans a year because I value the anatomy. Like if you're radiating, then is it close to the stomach wall? What is the approximation to the duodenum? What is the, vascular anatomy? That's sort of what we have been taught by the surgeons that we work with. So it's interesting. I don't know that there's a right answer, but when I see patients from India, there's a lot of PET scans. And I kind of wonder, how that it's also a lot of radiation, right? So I just wonder what, I don't know, Semanti, if you have any thoughts.
So, my reason to do the PET is mainly for the metabolic uptake and assessment for, following longitudinally the metabolic uptake and assessing response or lack of that based on the PET. I don't know how it is for Akash. For me, a lot of times, because just of the sheer volume we see, a PET and a metabolic image on the PET, and my team knows very clearly, very vehemently. I'm with show me the colored image. That has to be set out on the table when the patient walks in. And without that, I will not move the next step. It's very, very important and integral to the way I practice. So I think everybody has their own style. Yes, Akash, you're saying something.
Ma'am, so sir, I would agree we could have got a CT abdomen done and followed up. It it is just that if we get a baseline PET here, it is then gets easier to compare with PET rather than to ask a radiologist to compare it to PET. So it is just that that logistic is that she then was
No,
followed
you're
with
right. And
PET.
Akash, I think what Semanti said is absolutely your circumstance. I mean, I have the ability to look at every scan with the patient and look at before and after CT and review it. You don't have the time to do it.
No.
So perhaps PET is a better way for you. Yeah.
Because we have two things. We have the measurement and then you have the SUV uptake. With that, even we are able to make some sense of the radiology. And for other patients, even looking at the imaging ourselves, plus most of us are solid tumor oncologists or maybe all kinds of oncologists. So we are community oncologists. We are, going through all kinds of cases. And we may not have the luxury of discussing all images with our radiologists. So a lot of times we are just looking at the reports.
I
So
think one more
yes,
point there. I think when it comes to PET versus CT, the advantage of getting a single PET is also you don't, at least in our scenarios, we don't have to order a separate HRCT of the chest. I think because we're dealing with tumors which are locally advanced, which are quite voluminous, and the chances that they would have a distant met is quite high as opposed to very small early tumors. I think something that you would analogous to breast cancers or colonic cancers. And therefore, I think the chance of picking up something positive on a PET is much higher, maybe in our set of patients, as opposed to patients which may be in a different demography. So I think that's why we tend to use PET more often than purely a structural imaging.
So before we move over to the next case, I would like to,
Excuse me, can I make a comment?
yeah, please.
Yeah, I am Dr. Deepti from radiation oncologist from ILBS Hospital, New Delhi.
Thank you, Dr.
So,
Deep.
yeah, regarding this patient, if it's my view that if radiation therapy patient is not fit for consolidative radiation therapy in view of uh, abutting the duodenum and the colon. But yes, we can give and hypofractionated radiation therapy to a dose of 35 gray just for as an immunomodulator. So if we are combining the radiation therapy with durvalumab, then there will be a change in, we can just immunomodulate the tumor and there will be a better response. So adding something will be a better option instead of not giving at all.
Sure, that's a great point. One could consider
Sorry,
doing.
can I just get clarification there? So that's, you're saying to do that during at the time of that kind of oligo progression that was seen on the
No, ma'am.
durva.
After the completion of first, like GEMS,
I see.
when we have completed with GEMS with durvalumab, and at that time, we can take a review for the surgery. If surgery is not feasible, in that case, then we can give consolidated radiation therapy with the intent for immunomodulation of the tumor. Means if we are giving as a consolidative radiation therapy, we need to give a dose of up to 54 to 60 gray. In this patient, she is not fit to receive a dose of 60 gray. So if we give a dose of like 35 to 40 gray in this group of patients, then there will be an immunomodulation of the tumor microenvironment, which in turn results in the better outcome after durvalumab. Means we have seen such type of response in patients of intrahepatic cholangiocarcinoma, where we add radiation therapy followed by chemo and immunotherapy, resulting in good overall survival of median of 21 months.
So I think
Dr.
the jury is still out. I think Dr. Arman.
If I could have just a small word.
Yeah, please.
So wondering, and I would want everybody's word on it. So if we see a patient, any patient, not just this, with a look with say gallbladder with a large node, infiltrating duodenum, colon, node encircling portal vein. Say even if on scan we have a response, do we still consider them ever to be of a curative intent, maybe with any form of local therapy? So my understanding was if initially it is considered labeled inoperable, it continues to be in that forever. So if others could also pitch in,
Yeah,
I'm not sure.
Thank you, Akash. That's a brilliant question. I'll bring this question over to Dr. Shroff and Dr. Javle. As a wrap up, we are getting late for the next
Yeah, I
case.
think so. Akash, what you said was generally we never saw good responses with this disease, and it was very rare to ever make this patient who was completely operable. But this paradigm is changing because our treatments are working better, particularly our HER2-directed therapy. I have some patient with just Gemcis and Durva. So there are a few patients who can get there. And in fact, your own data from Tata with Rena Engineer, those patients would have been considered inoperable in America, but many of them did undergo surgery. So I think this paradigm is slowly shifting. So I think I want to give the other presenter time. So Sevanti,
Yeah, we'll move over to the next case. Thank you so much, Akash, for a wonderful case presentation. We now move over to Dr. Niyati Shah. Niyati is a young, talented, budding star at our center at Reliance Foundation Hospital. Niyati, thank you so much for making time today to present this case on cholangiocarcinoma, bridging borders with biliary cancer management. And we have the same group of panelists here as well. Dr. Prasad Dandekar, Dr. Saumil Vyas, Dr. Prashant Kumar, Dr. Dhaval Thakkar, and of course, the two experts, Dr. Rachna Shroff, and Dr. Milind Javle. Please go ahead, Niyati. The stage is yours.
Thank you, ma'am, for that introduction. In the interest of time, I'll start with the case directly. So I'm going to present a case of intrahepatic cholangiocarcinoma. Is my screen visible?
Yes.
Okay. I'm presenting a case of a 63-year-old gentleman. He was hypertensive and had hypercholesterolemia. He presented with deep-colored urine in January 2025. On examination, he had a performance status of 2. He had jaundice, mild hepatomegaly. Rest of the systemic examination was normal. His labs showed deranged liver functions with a total bilirubin of 9.6 and a direct bilirubin of 8.7. The remaining labs, renal parameters, and CBC were all within normal limits. So he was evaluated further to understand why he was having the hyperbilirubinemia. MRI abdomen was advised by his local physician, which demonstrated a lobulated mass in the perihilar lesion and caudate lobe of the liver. The lesion was encasing the left hepatic duct and confluence of the hepatic ducts, the common hepatic duct. It was causing compression of the IVC and intrahepatic portion with moderate dilatation of IHBR noted in both lobes. So with this finding, the patient went on to undergo a PTBD followed by SEM stenting in January. His liver functions gradually improved post the procedure. The bilirubin came down to 2.5 and so did the enzyme started stabilizing. He also underwent a biopsy of the hilar mass, which came back as intrahepatic cholangiocarcinoma. He went on to have a staging scan. A PET-CT was done, which showed a mass involving the perihilar region in segment 1, 7, 8, and 6 and 4A of the liver with an SUV of 5.5. It measured 6.5 into 7.5 centimeter, as described earlier. There were also some periportal, portocaval, and retroperitoneal lymph nodes, largest 1.4 centimeters. These are the images of his first PET-CT scan, where we can see the mass in relation to the other organs. So I've mentioned the questions along with the presentation. Maybe we can address them at the end. So what would be the frontline treatment offered for the patient? As we've already discussed the TOPAZ trial earlier, durvalumab, gemcitabine with platinum is considered as first line in advanced biliary tract cancers. We've seen improved overall response rate with the addition of durvalumab to the gemcitabine cisplatin backbone, with response rates going up to 26% as compared to 18% with gemcis alone. And of course, it comes with a PFS advantage as well. We've discussed this earlier in the previous talk by Dr. Shroff. So the patient was also offered the same regimen. He was started on durvalumab plus gemcitabine and cisplatin. We did a CT at the end of 2 cycles, which showed stable disease. The clinical course of his treatment was complicated with recurrent fever secondary to UTI. However, we managed to complete 4 cycles till May 2025. The CA19-9 trend was decreasing satisfactorily. We started off with more than 5,200 of CA19-9, which came down to 272 by May 2025. So at the end of So I would also like to pose this question to all the panelists that along with an advanced intrahepatic cholangiocarcinoma diagnosis, do you do parallel genomic testing for the patient while you start on the frontline therapy, or is the genomic testing reserved for once that the patient has progressed? This is the treatment algorithm for intrahepatic cholangiocarcinoma, ESMO guidelines. We've already seen and discussed this. A lot of newer targeted therapies are there in this field and many more to come. The molecular landscape of biliary tract cancers with FGFR2, IDH1, HER2, and other targetable mutations now, for which we have therapies and clinical trials available. We also did genomic analysis for this patient. A Garden 360 test was done on the biopsy, which revealed a IDH1 mutation. So coming to the imaging at the end of 4th cycle, A PET CT scan done at the end of fourth cycle showed slight increase in the metabolic activity in the primary mass, as well as increase in activity in the periportal, portocaval, and retroperitoneal lymph nodes. Now, the internal mammary and right supraclavicular nodes were also metabolically active, so it showed a progressive disease. This was the scan at the end of 4 cycles. A comparative image here to show the difference in uptake and increase in the disease. So any role of liver-directed therapy at this point of time in the form of TACE, TARE, or any role of radiation in the form of SBRT to control the liver disease, the patient continued to have a stable liver function test. But in any such progressions, if the patient does start having liver dysfunction, do we consider liver-directed therapy at this stage? Any alternate therapy options considering the high cost involved in using targeted therapy like tablet ivosidenib in India? So just a slide or two on the Claire Hardy trial where the ivosidenib was used in the second line setting. And this has already been discussed extensively previously. So I'm just going to go through this slide as well. Ivosidenib showed a median PFS benefit, 1.4 months versus 2.7 months. We have currently started the patient on irinotecan chemotherapy. The patient is doing well. He's been counseled regarding tablet ivosidenib and the cost involved. And the family is considering that option as well. So I'd like to hand over to Dr. Sevanti, Dr. Javle for taking it further. I want to thank Jarurat Care Foundation for giving me the opportunity to present the case. Thank you.
Niyati, thank you so much. And I would like to acknowledge that this is a case that truly has been managed shoulder to shoulder with Niyati and my team, and we feel challenged. And we feel challenged more so because we have patients who stretch themselves in affording this combination regimen. And so actually, with the questions that Niyati just presented, I'll bring the question over to Dr. Shroff here. This is a patient where we have offered frontline treatment with Gemcis Durva, and we saw that the patient has an IDH1 mutation. Would you have done anything differently? The patient had unresectable disease, hepatic artery involvement, and multiple lymph nodes. And so it was considered unresectable. The patient had this presentation where we started with palliative regimen, Gemcis and Durva. But would you do anything differently for this patient in frontline setting?
Yeah, no, I mean this is a great case and I think perfectly emblematic of the typical cases that we deal with, right? And that we see. No, I mean, I think in somebody that had the extensive disease that with the, local invasiveness of the disease as well as the lymphadenopathy, the palliative approach with Gemcis and immunotherapy, I think is very appropriate. And and that's regardless of knowing whether or not they have an IDH1 mutation up front versus even in the middle of that therapy. Gemcis, Durva, and I mean, it did help for a while, right? And so I mean, I think it is, while it doesn't always produce long-term control and or benefit, I think it is the standard way that I would have started.
Yeah. Thank you. Now that you have the study with the combination, when would you consider a patient for the combination study where Gemcis Durva plus ivosidenib has been offered?
Yeah, I mean, in a patient like this, I guess if the IDH mutation is discovered in time, it sounds like performance status was a little bit marginal, and assuming that the biliary drainage was allowed to, make the patient eligible for a clinical trial, right, between the performance status as well as the liver function tests. That study, again, just like most frontline studies, does allow for initiation of the Gemcis Durva and then subsequent enrollment. So, if there was access to that trial and the patient met the laboratory and PS parameters, I would offer the patient that trial. I don't know if Dr. Javle, if you feel differently, but
I agree with Rachna. So, Niyati, very nice job there. I want to say, let you know that, when we did that trial through another group called Accrue, you do see a lot of myelosuppression, when you combine ivosidenib with the others. Now, it's combined with leukemia all the time, right? They combine it with AML, but with donorubicin, but they don't worry about counts in that disease. So we learn, like Dr. Shroff said, we learn how safe it is to give it. I think your initial question, we try to profile everybody as soon as we can, not wait till the last moment. And I agree with you. These drugs are difficult to access. Ivosidenib is expensive. But this is sort of how it started for Dr. Shroff and I. We were profiling before we had drugs. And that's through that we learned. And eventually they will come. And, these companies like Servier is kindly sponsoring this meeting. They will help, in terms of hopefully providing some assistance to some patients, that sort of thing. So, but if we don't even look, we will never find any option.
Thank you. Thank you. Niyati, what was the biggest challenge as we were treating this patient? Would you like to highlight, you commented something regarding the ERCP or the PTBD.
Sure. So
Yeah.
I think so the initial challenge when the patient presented for the first time was they came with a very high bilirubin of more than 10. And we see this most commonly with a lot of patients who come to us with biliary tract cancers. By the time they have mild jaundice and they are getting evaluated, it's too late by the time they reach the oncologist, and generally the bilirubin levels are sky high. They generally require a procedure, some sort of ERCP procedure or PTBD procedure to get the bilirubin down, and we have to wait for some time before we can really start therapy. So that is the first challenge. And I think with the stenting and with in especially in elderly population, there are recurrent infections that we've seen. This gentleman also had recurrent infections, recurrent UTIs during the chemotherapy. That was the second challenge we faced while delivering the treatment for him. So I think combined all of these together, there were quite some treatment breaks and dose reductions that were required. And I think that did affect the outcome overall as well.
Thank you. Thank you. That's very that was quite a bit of an insight. I think Dr. Noel Thakkar is having some
Yes,
problem.
ma'am. No, ma'am. I've been able to
Okay,
join.
okay, okay. Thank you.
Yes, ma'am. Yes, ma'am. Thank you, ma'am.
So we asked the question of ERCP, PTBD with the last case and also with this case. How I guess you don't get to choose, you get called. But
No, yes, ma'am.
how do you participate in that decision making, as well as question regarding liver-directed therapy for cholangiocarcinoma, where what is the role of TARE in this setting? Not essentially for this patient, but in general, we wanted to learn from you.
So, ma'am, if I may say, generally, as we know, if the patient has intrahepatic biliary dilatation, we strongly consider ERCP to be the first line of therapy because it is not a percutaneous. You are not going from the liver capsule. It is less invasive, and the complication rates are less. But if in a case of a failed ERCP or we are suspecting a tumor around that area where we are not going to be able to negotiate, then we would go ahead with PTBD. The only two relative contraindications to PTBD is ascites and low platelet. We can drain the ascites and platelet and INR we can correct. Going forward for cholangiocarcinoma, as we speak, TARE, we have done for, there are few targeted therapies that we see. And one of the patients that we had done TARE, so the patient's last patient that the patient had come today, one of that patients, the patient had a good result for that matter.
Yeah.
And
So we do see certain cases where there is a localized disease and the entire play out is in that area where the assessment is done and treatment with TARE could be a possibility. Have you had situations where you've had to weigh in between tear versus, your colleague may be seeing the same patient for radiation.
Yes, yes, yes.
How do you make a case? And
So, ma'am, we have a clear guideline and indication. If for the patients with TACE, if the lesions are less than 3 centimeters, but if there is a large lesion, and so tears are always iffy as per criteria as we see BCLC criteria. These are RF ablations, they are definitive treatments. So tear and tase, as we speak, they are bridging therapies for, we are taking the patient from and giving time for liver transplant. And we have to add therapies with chemo. If we see for PET scan and if the disease is spread, we would go for targeted therapies for liver so that the tumor load is reduced, either by doing it RFA or we do microwave or we do TACE or we do TARE, and we take off the tumor load from the liver, be it any primary disease or secondary. And then if the disease is spread, then we would go ahead with doing the specific target of therapy of chemo for that matter. So then we do, it is more so of a multidisciplinary approach as we speak, not just a definitive one treatment. So for those cases where we can't give tear, we would do a lung fraction. We would see if there is a lung shunt more than 20-25%. The criteria we go with 25%. We do with macro aggregated albumin. We do a first scan to see if we give technetium 99, and they are the particles which mimic the actual yttrium 99. So we do that, and if we find the lung shunt to be more, or for cases where we are suspecting, for two of our patients last, where there was some of the flow going to the splenic artery and some of the flow going to pancreas, GDA, gastroduodenal artery at the duodenum. So we had gone and then super selective embolization for gastroepiploic artery, GDA. We have embolized a branch of splenic for that matter, and we have super selective. Even after that, we have super selectively gone. So how I do it? I divide the tear doses into four parts. If the four branches are being supplied, many people just inject the drug from the proximal, but I have a tendency we go as distal as possible from the microcatheter, dividing the dose according to their weight and the radiation, maximum radiation that we can give, 250 or 300 grays. And then as per the formula, we go as super selective as possible. And if there is any chance on the PET, we have found that the drug is going to go and fly and affect that particular area, we super selectively, not proximal, but super selectively embolize them and just give the targeted therapy to the tumor. And we have three cases where 12 centimeters lesion, 10 to 12 centimeters lesion, the entire lesion has disappeared in 4 to 6 weeks. With the AFP coming down to the values of 1 and 2 digit from 4 digits. So we have had very good results. Like,
thank you. Great, great. So I, first of all, congratulate you for the work that you're doing. And
that's
so
it. It's only under your guidance. It's only genuinely it's a team. And
a team player, I think the buzzword here is multidisciplinary care,
it's, yeah.
especially in patients who have disease that are difficult to control. And so thank you, Dhaval, for your
Thank you, ma'am.
input. I then come over, I
Can
think.
I make a comment? Can I make a comment?
Dr.
Again,
I will come back to you in just a couple of minutes. I have to ask questions to the panelists, which I would like to direct the question to Dr. Prashant Kumar here. Dr. Prashant, we just saw Dr. Rachna described the host of biomarkers, and even Dr. Niyati described the biomarkers that are involved in cholangiocarcinoma. So what's your take on when should we test and what should we test? How much should we test?
So I would say the biomarker, real use of biomarker, especially in intrahepatic cholangiocarcinoma, it has revolutionized Because why I'm telling you, because this is where we get multiple actionable mutations. And again, biomarkers which enabled us for the targeted therapy or immunotherapy, assisting diagnosis, prognosis, and monitoring. And I'll just give you example one after other, and which all of you maybe I'll able to convince you how the biomarkers played role. FGFR2 fusion 10 to 15%. There's a target. IDH1 10 to 20% cases. And again, ivosidenib doing miracle. Inacidenib, which is not approved for the cholangio, but for the leukemia, it has been approved. BRAF V600E, maybe probably 4 to 5%. HER2 amplification, 5 to 10 or 5 to 12%. Enter fusion, BRCA1-2, again PARP inhibitor may benefit, PIK3CA, which could be occasionally, and I think for those mutations we can target the pathways. So I would say that the future direction is, other than these biomarkers or the molecular biomarkers I mentioned, the ctDNA or epigenetic biomarker, microRNA or exosomes could be used. Depending on what you asked, it could be NGS covering the hotspot mutations, or the CGP could be used, where it could enable actually both DNA and RNA. So you can identify fusions also. Of course, ISC for HER2. So there are multiple options, especially in cholangiocarcinoma, and most of them are targetable.
Wonderful. Thank you. Thank you so much. I bring back to finally Dr. Somil Vyas. Somil, any specific thoughts? This case was advanced. We had a situation where hepatic artery was involved and multiple lymph nodes. Would there be a situation where you saw lymph nodes, but still you thought of a situation with resectability?
Yeah, so thank you, Dr. Seventy, for that question. Difficult case, always difficult to decide. I think at the outset, when the patient first presented, when Niyati presented, retroperitoneal nodes, clearly an M1 disease. And the question is, do you resect in the presence of retroperitoneal nodes? And I think when we saw the subsequent scan, there were nodes outside of the abdomen, right supraclavicular node, which clearly makes it unresectable. So I think, of course, if you got nodes outside of the abdomen, there's no discussion. It's M1, and one doesn't explore curative options. Now, if you got isolated retroperitoneal nodes, then the question is, do you really resect? So this is typically what is called as the station 16A node, the interaortocaval node. And if you got an isolated node between the aorta and the IVC, the patient has done well to neoadjuvant therapy, which I think here the patient did. You got your CA19-9 drop to 272 from 5,000 plus. Then, and let us make an assumption that there were no nodes outside of the abdomen and the patient was otherwise resectable, then having an isolated interaortocaval node essentially is not a contraindication. In fact, there is a very good paper coming out from the Rajiv Gandhi Institute of Surgical Oncology, which actually looked at this particular question. They sampled the 16A node, the interaortocaval node, sent it for frozen section. And if that was the only node positive, they went ahead and resected. And the oncological outcomes were much better than when you abandoned the procedure. So isolated 16 station 16A node in a patient who has otherwise got a good performance status has responded well to your chemotherapy. And this is where the role of a medical oncologist and a precision oncologist like you comes into play, because you make more patients convertible than which would otherwise not have been convertible. Then there may be a role for carrying out selective extended lymphadenectomy after due discussion, weighing in the risks and the benefits, and making it safe for the patient and ensuring that you get an R0 clearance. Yeah. If you got nodes on the left side of the aorta around the renal vein, because the renal vein would straddle around there, then obviously that would become a bit too much and you would call it off. So I think this is one of the few exceptional situations where you would offer a resection. The second situation where you may offer a resection to a patient is, very often we do see that they get isolated satellite nodules on the side of the liver that is going to come out. So you got a gallbladder cancer, you got a satellite nodule or a intrahepatic cholangio in, let's say, segment 5, and you got a nodule straddling in segment 7 or segment 8. Technically, it is M1, but it's still lying in that part of the liver which is going to come out. So this may be another case situation where you could offer a potentially curative resection in a patient who otherwise has M1 disease. So these are the two situations where I think one should look for resecting in patients who can go through a resection with a margin of safety.
Great answers. Thank you so much. I think the bottom line that I get after what Dr. Somil said, of course, this patient was not in that category. The patient had advanced disease, very high bilirubin and nodal positivity, hepatic artery involvement. But the bottom line from what Dr. Somil said, what we heard from others, including Dhaval, is that we should look for multidisciplinary care. Prasad, last words from you regarding this case. Currently, you saw the imaging that Niyati showed. Any role for radiation in management of intrahepatic or cholangiocarcinoma in general?
I think Dr. Prasad has dropped out of the call. He dropped.
Okay, no problem.
Maybe Dr. Deepti can answer the questions because
Yes, sure. I was going to come to Dr. Deepti now.
Yeah,
Yes.
hello, ma'am. So again, in this patient, my take will be to start with radiation therapy. We can give radiation therapy in hyperfractionated or SBRT form, which can be completed within a span of 10 days. By that time, your biopsy and your NGS will be coming. So radiation therapy followed by chemotherapy plus immunotherapy. So there's a good paper by there's one paper by I think published in 2019 yeah, by Yakashita et al., which have found that if the local disease can be controlled, it results in improvement in the cholangitis. There will be decrease in the chances of cholangitis and mortality morbidity, leading in such group of patients, resulting in improved overall survival. So even if the patient has metastasis at the baseline, so one need to take care of the hepatic disease. So that hepatic disease can be taken care by the surgery or any of the loco-regional therapy. Now in this patient, because lymph nodes are also present, so one can deliver radiation therapy to the involved lymph node along with the liver lesion, resulting in local control, followed by chemotherapy, which in turn results in the take care of micrometastasis, increasing in overall survival and PFS.
Excellent.
So we usually treat like this only, and we have a recent publication on role of radiation therapy, SBRT, in locally advanced or metastatic intrahepatic cholangiocarcinoma.
Thank you. That was great insights. I think in interest of time, we will wrap up the case. But for final words, would you manage anything differently? Dr. Shroff, Dr. Javle, you're all with me on this? Yeah, okay, great. So thank you, Niyati, one more time for a great case, and all our panelists for very, very engaging discussion. Thank you, Akash, for your case as well. And thank you, Priyanka, Jarurat Care. Thank you, Dr. Javle. Thank you, Dr. Rachna Shroff, for brilliant presentation. Back to you, Priyanka, to take this forward from here.
Thank you everyone. It was a very insightful session. I mean, half of the things were beyond my understanding capability, but I did try to understand, and this is really what I wanted, to have a multidisciplinary discussion. And that is the agenda of Horizon, to bring in, bridging the borders between Western sciences and also discussing more about the multidisciplinary care. So I'm very grateful for everyone to participate in the discussion. And I'm also thankful to the sponsors AstraZeneca and Servier to help us with the entire their consistent support in bringing together this organization. I think I'll give it to Dr. Javle to take it forward now. Thank you.
Yeah, I just again want to thank Rachna for just a fantastic talk, and to all of you for, each time we Indians get together, we have a lot to say. So I'm just very happy that it was very interactive. We have a great lineup coming up. We have a talk on immunology by Sangeeta Goswami, just immunology in cancer. Really a fantastic young scientist like Dr. Shroff. And then we have Mitesh Borad, who is a good friend of ours and leads the HCC program at Mayo Clinic. He's coming up as well. And these will be a couple of talks, but bring in some suggestions. The focus is really hepatobiliary, as well as, general management of cancer as well. So if you have any thoughts, suggestions, do let Priyanka know. And thank you so much for joining us this evening in India.
Just one last request, if people can try to turn on their camera, we can take a quick picture and then we can call it a day. Yeah, great. All right. Yes, 1 second, okay, I'll do it in a count of 3, 2, 1. Yeah. All right. Yeah, thank you. Is there anything else pending, Dr. Sevanti, Dr. Javle, Dr. Shroff, from your side to add?
Just that this is going to be a monthly meeting, and we need to all be energized to join in and contribute, and we're looking for feedbacks and let us know how to make this better. But congratulations, Priyanka, on your
Thank you
first one.
everyone for the support.
And thank you, Dr. Shroff. Thank you, Dr.
Thank you
Jha.
very much, Dr. Shroff. Yeah.
Well,
Right. Thank you. Have a great weekend. Bye-bye.
Thank you all.
Thank you. Thank you very much.